Hello, Eastview Scholars. Welcome to this edition of Sharp Shorts, where we'll talk about how cells talk to each other. And here's the reality. Whether you're a single cell or a multicellular organism, there needs to be some communication, either between the group, as we saw with bacteria and discussed with quorum sensing, because one little microscopic bacteria isn't going to do anything or have any effect on the organism that it is using as a host, but a group of them will. And of course, for multicellular organisms, you need some way for all those cells and systems to talk to each other. And that always comes down to a chemical signaling question. So that's what we're looking at, how they talk together to coordinate. Now, if you are multicellular and your cells are stuck together, as we can see here with plants or attached to each other, as we can see with plants and animals, then you have gap junctions or plasmodesmata, and you can those signals can easily transverse through the cell membrane and through the gap junctions that are there. Or if they're close to each other, some cell-to-cell recognition like that. There's also local regulators. If you can see here with paracrine and synaptic signaling, like you would use here in your nervous system, the release of the neurotransmitter or whatever the target chemical is, receptor signaling molecule is in a small localized area. If the cells are not distant, and this is for you while you have a hormonal system, where those hormones, those chemicals are then released throughout the blood and they travel and they come in contact with every single cell, but they only have an effect on the ones that have the appropriate receptor. Okay. How do we look into some of this? Let's just look quickly at yeast and the mating types of yeast. What we figured out is yeast have two types of cells, mating type A and mating type alpha, of which they have receptors on their outside to recognize the mating factor that the other one produces. So here this produces that. Notice it fits exactly in that receptor. Here's the mating factor of A. It fits exactly in that receptor. So when they release those factors and that cell nearby binds that, then that's the signal for them to mate. And then you get a new combined yeast cell with a mixture of DNA. And that is one of the things we'll talk about later with variety and changes in the habits of different sexual lifestyles of different organisms, is how do they keep variation, especially single cell organisms, how do they keep variety? It can all be through mutation. Anyhow, really what we're looking at is there's three parts to cell signaling. The reception part, which we just looked at. The transduction part, which is here. Signal transduction pathways are mechanisms for looking at or advancing that signal until you get a response, which is the third thing. The reason this will be an important view in our next film, this film will talk about reception, is what it can do to amplify signals, so you don't need a large mass of chemicals to make it work. Anyhow, so that's the three steps. Some signaling molecule, some ligand comes in, creates a change, and all these changes we'll talk about with reception and transduction, these are proteins. And so the changes are often conformational changes in the structure that activate another response. But you have this reception, you then have the transduction of the pathway, and then you have whatever the cellular response could be, and that could be anything. Okay? So let's talk about reception in this film. They can be intracellular right on the plasma membrane, as you saw in this picture, where it binds and you have a protein that spans the membrane, or it can be internal. Okay, so that if it was a small hydrophobic molecule that could pass through the cell membrane, it could meet the cell receptor internally, the receptor cell internally. As you can see, most water-soluble signal molecules bind to specific sites on those membrane proteins, and the three main types are G protein, which is the largest, tyrosine kinases, and ion channel receptors. Let's just go through them quickly because you can read about them. G protein receptors are the largest. You can see this very complex. Here is where the signaling molecule would come in, and then you'd get a response on the other side. It's an on-off switch. Okay, and let's just go to the picture and see. So here's what we have, and here's the basic setup. Here's expanding the membrane. So here's the receptor protein. Here's the GDP in its inactive state, and here's the enzyme that's going to help coordinate the transduction. Our signaling molecule came in specific for that G protein. That then changed its conformational shape, which drew an attraction to this inactive. GTP comes in, GDP is out. These are energy molecules. And when that happens, there's the change in that, and then it's much more attractive to activate the enzyme, and it will diffuse across a short distance. Activate the enzyme, which will have a transduction pathway and a cellular response, at which point the enzyme will also then dephosphorylate, and the GDP becomes inactive again, but is ready to go through again. Okay. Another is receptor tyrosine kinases. They are different than G proteins, but their ability is that they can activate more than one complex. So they really can amplify a signal or get more than one action going on. ACE means enzyme. Kinase is an enzyme that catalyzes the transfer of phosphate groups. So these change things into higher potential energy molecules with adding a phosphate. Okay? And here what you can see, oftentimes here's the inactive form, and what you have here is you see all the tyrosine, the amino acids here. Here's the signal molecule coming in. Now you need two of them to bind, and they form what's called a dimer. That's very common in the tyrosine kinases. Let's go back. That dimer formation activates those regions that are unphosphorylated. It's very easy then with ATP to phosphorylate them. You now have fully activated, and you can trigger multiple cellular responses once you have activated that dimer. You can see the enzymes coming in with the phosphorylated dimer triggering those responses. So the real benefit is the multiple responses you can trigger. Okay. Ligand-gated ion channels, these can be chemically gated, electrically gated, certain different ways to go, but you can notice that it's closed, and then the signal comes in, it opens the gate, it allows for diffusion of whatever that molecule might be. This is one way that the nervous system and neurotransmitters work, allows them to diffuse in and flood in, and then based on the levels, again, then the gate will close and the molecule will be released. Intracellular as opposed to intramembranous, these are the small hydrophobic that can pass through, like steroids and thyroid hormones, and activate hormone receptors and stuff that could be activators of transcription, turning on specific genes. Here we'll show you one here. Here's testosterone coming in. You can see what happens. Testosterone is a steroid, very easy to go through the cell membrane. It finds its receptor complex internally. That binding allows it to go through the nuclear pore, otherwise it could not. That triggers the transcription factor. Okay? Binds to the specific gene it wants to have copied to make whatever protein it needs to make in this case. Stimulates transcription of the gene in mRNA, which we'll talk about in the future. And that travels out, the mRNA messenger RNA travels out, and you make a new protein. Anyhow, that's it. But that's just the first start of what we're going to do in our communication, and that's the reception. You have some big intramembranous protein ones, and then for those signaling molecules that can pass through the membrane, you also have some intracellular targets. Anyhow, that'll do for now. Our next will be the transduction pathway. Scholar on, Eastview.